students, welcome to Biosastra Talks. I hope all of you are doing well and preparing for the NEET exam as well. And all the best for every one of you. And as we already know that we are going on with the NEET topic-wise paper revision. In the previous videos, we learned about the process of photosynthesis and how the process steps will take place. In this also, we are going to know about one of the important topic from the photosynthesis, that is nothing but C4 pathway. Okay, as we already know, this is also one of the important topic from NEET, through which recently repeated questions were coming. Okay, so if you see what is a C4 pathway, it is also known as a Hatch and Slack pathway. Okay, why? Because these two scientists invented this pathway, C4 pathway. Normally we know that we know that C3 cycle. So what is a C3 cycle? It occurs in C3 plants. Okay, so like that means in same C3 and C4 are similar. Both of them will help in the fixation of carbon, but in a different way. Okay, so alternative for this CO2 fixation mechanism found in some plants, that is nothing but we know that there are so many types of plants which will survive under different conditions. Okay, like that if you see that some of the plants undergo the C3 cycle, whereas some of them which cannot survive in the low temperatures, means that the plants which can survive in the high temperatures, intense light, like you can also say this is a desert plants, cactus or maize. So these are all can undergo this C4 pathway. Okay, so and normally this was discovered by the scientists Hatch and Slack. That's why it is also known as Hatch and Slack pathway. So what was the first, why it is known as C4 pathway? Means like in the C3 cycle we are having the first first compound, that is 3 carbon. Like in this pathway also, the first stable product that is formed is nothing but a 4 carbon compound. Well, it's containing 4 carbons, that's why it is known as C4 pathway. Okay, so that 4th carbon compound is nothing but oxaloacetic acid. So okay, so this is the C4 pathway, why it is named and everything. And here you can see some of the examples of C4 plants like sugarcane, maize, sorghum. Means these all plants can survive in the higher temperatures like arid, semi-arid tropics or like that. So in these plants we can observe the C4 pathway when compared to the C3. Okay, so normally these are the some of the examples of C4 pathway. Okay, let's get into the topic, and now we can see what is the main difference between the C3 and C4 plants actually. First thing is nothing but the main difference between the C4 and C3 plants you can see is nothing but a Kranz anatomy in the leaf structure. Okay, so what is the Kranz anatomy? Means this is discovered by the scientist. Okay, Kranz. So why it is known as Kranz Academy? Kranz means breadth, means normally in the C3 plants you can see the normal order of the normal arrangement of the cells and everything, but in here in the C4 plants we can see some type of breadth type of like clumsiness and everything in the leaf structure. Here you can see the structure here, upper epidermis, lower epidermis. In between normally we'll have the mesophyll cells and everything, but in the C4 cycle or C4 plants we can also observe the bundle sheath cells present inside, means which were surrounded by the mesophyll cells. Okay, so like that means this is the main difference between the C3 and C4 plants. Normally C3 plants does not contain these cells. Okay, so like that. So two types of cells are present, that is mesophyll cells, and second one is bundle sheath cells. So the process takes place in both these cells in the C4 plants. Okay, and bundle sheath cells, if we compare these C cells, bundle sheath cells are large, thick and lacks intercellular spaces. Why? Because here you can see, but whereas when compared to the mesophyll cells, they can arrange easily and they'll have more intercellular spaces, whereas the bundle sheath cells will not have intercellular spaces. And when compared to the mesophyll cells, they also have the large chloroplasts. Okay, so like that. So chloroplasts are present in both of them, that's why they can undergo the C3 cycle, right? Photosynthesis. So like this is the main difference between the C3 and C4. And here you can see by the image how if you see the leaf cross sections, obviously you can understand. Here you can see, if we see the leaf cross sections here, this is the C3 plant leaf cross section, in which you can see that this is the stroma, and these are the mesophyll cells and bundle sheath cells. Okay, in the C3 plants also you can see both the cells, but but they are arranged in an order, like in an order in which you can see, like here the mesophyll cells are present here, whereas the bundle sheath cells are present here. Okay, means you can differentiate them easily in the C3 plants, whereas when we come to the C4 plants, that's why it is known as a wreath, means clumsy. Why? Because here you can see, so these are all the mesophyll cells which were surrounding the bundle sheath cells here, right? Means the bundle sheath cells are present inside the mesophyll cells. Okay, so like this you can see that the clumsy nature and wreath nature in the C4 plants. So this special arrangement or anatomy is seen in the C4 plants, that is known as Kranz anatomy. Okay, if you compare both of them, that's why in the that's why the carbon fixation in C3 and C4 plants is somewhat different. Okay, so normally in the C3 plants, the carbon fixation occurs in the stroma and mesophyll cells, right? Whereas in the C4 plant, you can see the carbon fixation, means C4 pathway in both mesophyll and bundle sheath cells. Okay, this is the main difference, right? So if so here, if we go on to the next process, that is nothing but the formation, like what were the steps included in the C4 pathway. So what were the main steps that were included in the C4 pathway are normally 5, likewise in the C3 cycle. So what was that was, first one is CO2 fixation in the mesophyll cells. Obviously, we know that here the C4 plants contain both mesophyll and bundle sheath cells, and it occurs in both the cells. So first of all, the CO2 fixation, it starts from the mesophyll cells itself. So what happens in this fixation is that the atmospheric CO2, the CO2 which enters these mesophyll cells, combines with the phosphoenol pyruvate of the mesophyll cells in the presence of PEP carboxylase enzyme. It undergoes certain like breakdowns and form a 4-carbon compound, that is nothing but oxaloacetic acid. So that's why it is known as a C4 pathway. Obviously, we learned that the first product that is formed from the C4 pathway is nothing but a 4-carbon one. That's why it is known as C4 pathway. So if we see it in an equation way, here you can observe that the phosphoenol pyruvate is a 3-carbon compound. When it combines with the carbon dioxide, it forms a 4-carbon compound, nothing but oxaloacetic acid. Okay, so if we move on to the next step, what is the next step? That is nothing but formation of malate or aspartate from this oxaloacetic acid. So what happens here is the oxaloacetic acid combines with the NADPH molecules in the presence of protons to form malate and aspartate molecules. Means both of them are 4-carbon compounds only. Here no removal of carbon dioxide or anything, okay, or addition or removal. But here the 4-carbon compound again converts into 4-carbon compounds with the help of some energy which is released from the light reaction process. Okay, here if you see in an aquatic way, like equation way, this is the reaction. The C4H like oxaloacetic acid combines with the NADPH to form C4H6O5. Okay, here you can see the removal of oxygen atoms. So that oxygen is also evolved here in this C4 pathway. Okay, so like this oxygen means not in the form of oxygen. Here you can see that NADP plus no means those hydrogen ions combine with the free atoms to form water molecules. Okay, which were helpful for the plant to survive. So like this it happens in the second step, whereas in the third step the malate, here we got the two products, no, like malate and aspartate, both of them are similar actually. So in these two molecules only malate enters into the next process. Why? Because this aspartate is not needed here in the C4 pathway. Only malate enters the next step, that is nothing but it is transformed from the mesophyll cells to bundle sheath cells. Okay, so like the malate enters these 4-carbon compound transported to the bundle sheath cells which are present inside. If we see what is the next step, fourth step is decarboxylation in the bundle sheath cells. So here what will happen is the malate molecules which entered these bundle sheath cells were broken down, and the CO2 enters the Calvin cycle here only. Means the CO2 which is released from the malate breakage, like So it enters into the C3 cycle. Why? Because we know that the C3 cycle occurs in the bundle sheath cells only. That's why it enters the C3 cycle. And also you can see the Rubisco enzyme that fixes the CO2 into the Calvin cycle here. So this all takes place in the decarboxylation of bundle sheath cells. Okay, what will happen here in the aquatic way? Like if you see that malate molecule is converted into pyruvate, that is nothing but a 3-carbon compound. Means here you can see removal of carbon dioxide. How? C3H4 H6O5 means here C3H4O3 is formed, means one carbon and oxygen atoms are released along with the hydrogen ions. In that, carbon and oxygen combine to form a carbon dioxide which enters the C3 cycle in the bundle sheath cells. Okay, so like this overall process takes place in the C4 pathway in the fourth step. Okay, if we move on to the next step, that is nothing but regeneration here. So the regeneration of PEP means here how again the PEP is formed. Why? Because we are saying it is a cycle, no? Like in the C3 cycle, again from the step ribulose bisphosphate is formed. Like that only in this cycle also you can see the formation of PEP from these pyruvate molecules. Okay, so what will happen is this pyruvate again moves down. Here you can see, first of all, the molecules are present in the mesophyll cells. They enter the bundle sheath, and again the pyruvate which is formed in the bundle sheath cells move on to the mesophyll cells. Okay, so like this it happens. So it is regenerated to PEP by utilizing some of the energy. Like if you see these pyruvate molecules with the help of the adenosine triphosphate, they form the PEP, means phosphoenol pyruvate molecules, by using the some of the like phosphate ions in the form of ATP. Okay, so like this the whole process will occur in the C4 pathway. Okay, like this in the C4 pathway, obviously you can see that the formation of water molecules, carbon dioxide, and thus the carbon dioxide which is evolved in this C4 pathway obviously helpful for the C3 pathway which occurs in the bundle sheath cells itself. Okay, so if we see for the to understand easily, here you can see what happens. Okay, if we compare it, so can you can easily understand. So what is that is, first of all, if we see that there are two types of cells, those are nothing but mesophyll cells and bundle sheath cells present in the chloroplast, like they contain the chloroplast. Okay, so here the C4 cycle occurs in the both of the cells. How it will take place, we'll see. First of all, like we learned in the first step, the phosphoenol pyruvate with the help of the carbon dioxide that is released into the mesophyll cells is converted into oxaloacetate molecules here. So means this is the first step that occurs in the C4 pathway of mesophyll cells. Okay, so what will happen, this oxaloacetate is again converted into malate and then aspartate, right? So why I have given arrows like this means they are like they can do vice versa. Means sometimes when the aspartate concentration is more, it can again convert into oxaloacetate or like that. That's why, means why, because both of them are 4-carbon compounds only. So here you can see the reversible reactions in this step. Okay, so obviously we know that this process occurs with the help of NADP molecules. Okay, with the help of the NADP molecules that were evolved. So obviously in the next step what will happen, these malate molecules which were formed in the mesophyll cells, they will enter into the bundle sheath cells. Okay, so it means they are transported into the bundle sheath cells here. Okay, so this transported malate is converted into pyruvate molecules with the help of some enzymes. So this what will happen, and it also evolves some carbon dioxide which is helpful for the C3 cycle within the bundle sheath cell. So how it will happen is, if you see that the RUBP in the C3 cycle, we can see that the RUBP with the help of the CO2, it forms a 6-carbon intermediate, and then it forms a phosphoglyceric acid, and again the process will take in place like this, means cycle and cycle. So like this the C3 cycle occurs within the bundle sheath cells with the help of some of the carbon dioxide that is evolved from the C4 pathway. So like this it will happen, and you can also see the formation of some of carbohydrates, that is nothing but glucose, okay, in this process. So here you can see in the bundle sheath cells, you can see both the C3 and C4 pathways that were undergone step by step, okay. So and then obviously the CO2 that is released is used in the C3 cycle. So what about the pyruvate here? So pyruvate, which is formed in the bundle sheath cells of the C4 plants, obviously it again enters into the bundle like mesophyll cells of the C3 plants, okay, sorry, C4 plants. So these pyruvate molecules, what they will do, they again converted into phosphoenol pyruvate molecules, means here you can see, so like it is in a cyclic way. If you leave it here, you can see that the molecules are moving in a cyclic way. It is a cycle, no? That's why this pyruvate which enters the mesophyll cells again, it is converted into phosphoenol pyruvate with the help of ATP molecules here. So from this whole cycle, you can see that obviously the energy molecules are needed for the C3 and C4 cycle in the form of ATP and NADP molecules that were evolved from the light reaction. So this is the whole process that occurs in the C4 cycle in the mesophyll cells and bundle sheath cells. Obviously mesophyll cells are present upper and then bundle sheaths are inside. So for the clear observation, I have drawn it like this, okay. So this is the whole process that occurs, and again and again it will occur like this only with the help of the CO2 and some of the enzymes, okay. So if we move on to the next one, so how, if we see the total reaction, how it will take place and everything, here in the mesophyll cells you can see CO2 plus PAP gives rise to oxaloacetic acid and then malate, okay. Here, and if you want to differentiate, means which steps will occur in the mesophyll cells and which steps will occur in the bundle sheath cells. In the mesophyll you can see CO2 undergo like combines with the PEP to form oxaloacetic acid and then to the malate. So whereas in the bundle sheath cells, this malate is converted into CO2 and pyruvate. So this is the overall process occurs, and then again this pyruvate enters and phosphoenol pyruvate is obviously formed in the mesophyll cells itself. So overall simplified summary, if you see that CO2, ATP and NADPH gives rise to glucose, ADP and NADPH. Why? Because this is a overall, like it is a, you can say that it is the for equation for the photosynthesis. Why? Because with the help of the CO2 itself only, obviously carbon dioxide is formed with the help of all these another molecules, okay. This is the overall simplified summary for the C3 or C4, okay. So like this it will happen. If we compare, means you should know the differences between the C3 and C4. No, what are the main differences? Some of the differences are, if you see in the C3 cycle, the ETP used per one carbon dioxide evolution is 3, whereas in the C4 cycle 5 ATP are utilized. Means the C4 plants need more energy when compared to the C3 plants, okay. Means 3 for Kelvin. Why? Here you can also see why. Why? Because in the C4 plants you can see both C3 and C4 like Kelvin cycle and Hatch-and-Slack pathway, whereas in the C3 plants you can only observe the C3 cycle. That's why it needed less energy, whereas the C4 plants need more energy, for 3 for Kelvin cycle and 2 for PEP regeneration, okay. So another difference is nothing but for the direct the C3 cycle is used for the direct CO2 fixation, whereas the C4 cycle it costs higher, means but it avoids photorespiration, but it needs more energy. That's why it cannot fix the carbon dioxide directly, because it needs more energy when compared to the C4 cycle, sorry, C3 cycle, okay. These are the main differences for the energy cost comparison, means energy molecules and everything. So energy requirement, if we see it in a number way, like how the molecule C4 pathway consumes more ATP when compared to C3. Here if we see, C3 plants need 18 ATP to form one glucose molecule, whereas the C4 plants need 30 ATP for the evolution of one glucose molecules. Means why? Because the C4 plants have to undergo both C3 and C4. That's why they need more energy for the formation of one glucose molecules here. So this is the main comparison between the C3 and C4 molecules. Okay, so this is the overall reaction and how it will takes place, and these are the main differences between C3 and C4 pathways. So obviously we'll move on to the next one, that is nothing but PYQs. So if we know the PYQs, and you can know the questions, like the format of the questions. So what was the first question? It came in the NEET 2021 paper. So that is nothing but C4 plants are more efficient in photosynthesis than C3 plants due to. Why? Normally C4 plants are more efficient. Why than C3? Why? Because presence of large number of chloroplasts, ability to fix more nitrogen. No, this is long. Lack of photorespiration and higher stomatal conductance. Okay, so these are the four options they have given. Here you can remove some of the options, like the presence of large number of chloroplasts. No, why? Because both of them will have that like that only, so this is not the main reason. Whereas the ability to fix nitrogen, you can see that there is no nitrogen fixation in the like photosynthesis process. Okay, and the next one is a lack of photorespiration. You can consider this option. Whereas the higher, and another one is higher stomatal conductance. Higher stomatal conductance will obviously occur in both of them, but this is not the reason for the efficiency. Okay, so here if you see, so what is the correct answer here is lack of photorespiration. Why? Because normally C4 plants does not show photorespiration. That's why they are more efficient in photosynthesis, but they need more energy, but they don't show photorespiration. That's why they are more efficient than the C3 plants here. Okay, so if we move on to the next question, which of the following statements is true about PEP carboxylase enzyme? Here they are asking what is the main thing, like what is the statement that is true about the PEP carboxylase enzyme. First thing, found in bundle sheath cells of C4 plants. If you see PEP is like formed in the mesophyll cells of the C4 plants, right? So this is not the correct answer. It is not found in the bundle sheath cells. It found in the mesophyll cells of the C4 plants. Okay, what is the second option? It is having more affinity for the oxygen. And the third one is it is the primary CO2 acceptor in the C4 plants. Obviously it is the primary CO2 acceptor. Obviously this is the primary CO2 acceptor, and it is true. Whereas it is involved in photorespiration. If we see from here, it is not involved in the photorespiration. So this is not the correct answer, and it is not having affinity for the O2. Why? Because the phosphoenol carboxylase fixes the CO2 itself, not oxygen. So this is also not the right answer. Obviously, what was the right answer is the primary CO2 acceptor in the C4 plants. Okay, why? Because the phosphoenol carboxylase, obviously it accepts the carbon dioxide from the atmosphere and it fixes into the oxaloacetate. Okay, so like that, this is the primary one. That's why it is correct about the PEP carboxylase enzyme. Okay, so we'll move on to the next question here. What was the question they have asked is which one of the following is the correct sequence of the steps in C4 pathway? Means here they are asking, oh, you have to say the order of the C4 pathway. Okay, first one is CO2 fixation, second then to formation of malate, and then to decarboxylation, and then Calvin cycle. Okay, this is the first option. If we move on to the second option, Calvin cycle, decarboxylation, malate formation, CO2 fixation. From the option itself, from the starting, you can say that this is the wrong one. Why? Because the Calvin cycle will not start from the starting. Okay, it comes in the middle. So this is the wrong answer. Okay, if we move on to the next option, obviously first CO2 fixation, and then Calvin cycle, and then malate formation, and then decarboxylation. And again in this option also you can differentiate. Why? Because first the malate is formed in the mesophyll cells, and then it is transferred into the bundle sheath cells, and it evolves the carbon dioxide which enters the Calvin cycle. Means it occurs in this way, means first malate formation and then CO Calvin cycle. So this is also the wrong answer. If we move on to the next option, first one, formation of pyruvate, and then malate, and then CO2 fixation, and then Calvin cycle. This is obviously the reverse one. Why? Because from first carbon is fixed, and then malate is formed, and then Calvin cycle occurs, and then pyruvate is formed. So this is also the wrong sequence of the C4 cycle. Obviously, what is the right one here? First one means, first of all, the carbon dioxide is fixed with the POP, carboxylase enzyme here. Okay, and then and you can see the formation of malate from the oxaloacetic acid, and those malate molecules will undergo decarboxylation to form pyruvate, and then with the formation of the pyruvate itself, it also undergoes the Calvin cycle. So this is the right order of the C4 pathway here. So like this, if you see the options, I'm saying it again and again, when you see any question, when you read any question, first of all, you have to consider the options, in which obviously you'll have one or two options which are not obviously suitable for the question. Okay, so like this, if you compare, you can easily answer the question. Okay, so these are the some of the PYQs that were came in the previous papers, and that's it, guys. This is all about the C4 cycle and some of the important topics and some PYQs from this topic, and I hope all of you understood well. And if you have any doubts, you can comment me in the comment section. And along with that, I have to make some announcement. That is nothing but now we are having only like less time for the NEET topics and NEET revision. We are moving on to the FAQs. Okay, this is the big announcement here from the today onwards, like till the exam, these 4 days we'll have some of the important tips and some FAQs, frequently asked questions, so that you can see the videos and you can revise the questions. Why? Because we are having the less time, you can obviously remind all these questions. So in the next video, I am giving you some of the frequently asked questions from the NEET exam, so that you can understand the topics and the questions as well. Okay, thank you, guys. Thank you so much, and we'll meet in the next video. Thank you.